Super. Well, thank you to the organizers and thank you to everybody here for attending. And I'm excited to be back and happy to talk about some advances that we've made in terms of our platform development, as well as our lead program, our small molecule STING agonist, and then some thoughts on future directions that we're taking things and methods to really go beyond binding and think about conformational modulation in the context of drug discovery. So a little bit about Silicon Therapeutics. We're a fully integrated physics-driven drug discovery company. We like to say that we have the whole pipeline from chip to clinic. We own our own supercomputer cluster of over 400 GPUs, lots of CPUs. We can run on all the different cloud platforms, and we're developing a custom supercomputer using field programmable gate arrays. It's a kind of an advanced hardware technology. We've developed a software platform based on quantum physics using quantum mechanical calculations to derive high quality force field parameters, and then a suite of molecular dynamics and simulation tools to explore things like binding, but also go beyond binding to look at conformational dynamics and conformational modulation. We've built out a team of experts that include experienced drug discovery scientists, as well as computational physics and software engineers. All of this is under one roof. We have about 20,000 square feet in the Boston Seaport area, with half of that being lab and half of that being office space. Everybody really combined with teams of biologists, chemists, biophysicists, and simulation experts all sitting together. We're in the process of transitioning to the clinic with our first in human hopefully coming soon, and going to be happy to tell you about the status of that STING small molecule agonist project here. So we've really connected the pieces needed to go around this full cycle, from the hardware and infrastructure with our cluster, as I mentioned, accurate molecular simulations, which includes a free energy simulation infrastructure and a high quality quantum mechanically derived force field. We've developed molecular design applications where the rubber hits the road in terms of simulations impacting decisions in drug discovery. And we typically go beyond just simple calculations where you produce a number, but we're really trying to provide insights to the design team. And that means a lot of molecular visualizations and representations of the underlying physics mapped onto the chemical structures and the proteins. And then more recently, we've been developing this targeted conformational modulation approach, where we're looking at not just the binding itself, but what happens after binding. How is the protein changing in terms of conformation and stabilization, and how is that connected to the underlying biology? And finally, that all gets connected through this integrated platform where we include biophysics and other experimental techniques, which I'll be talking about. We have a great team, really spanning from the theory and software side, with our CTO Huafeng Zhu, myself focused on the software and methods development, which then connects to the R&D team headed up by Chris Winter, Chris Borella heading up our preclinical and operations work, and then Humphrey Gardner, our CMO, heading up clinical, as well as Pam Carroll, our CBO, extremely experienced research and R&D person who's now migrated into a business role, and our CEO, Lanny Sun. Just a quick little shot of our spot in the Boston Seaport for people who have been there before. Often you will see our building when we get back to conferences. We're on D Street, right across from the convention center, the main Boston Convention Center. So great location, great lab space. We've just expanded to about 10,000 square feet of new lab space, and really excited to expand our operations. So I'm just going to show a quick movie here for people that are not familiar with molecular dynamics and molecular simulations, to give a sense of what we're talking about here. So what we're talking about is modeling the underlying protein at the most fundamental level, the atomic level. And that means including all of the biologically relevant components of the system, the waters, cofactors, and everything that matters. So here we're just stripping away those waters to get a sense of what's happening with the protein. What we care about really is how a small molecule binds, and you see the loop on the left, how that actually moves upon binding, because that's where signaling comes from, especially when we have more complex proteins. And then the question is, how do we modify this small molecule to impact binding and also impact the protein conformational state? Now, we can't actually predict what this conformational change is going to have on the underlying biology, because biology is extremely complex. But what we can do is come up with physics-driven hypotheses, which we can then iterate with the experimental data, both biophysics and biological data, in order to come up with these conformational hypotheses that then drive our drug discovery project decisions. Now, why are molecular dynamics simulations important, and why are people hearing more and more about these in the news? A lot of it has to do with technology breakthroughs that have come over the last 10 years, and most specifically, graphics processing units, or GPUs, that have allowed us to move from the kind of hundreds of picosecond timescale per day to hundreds of nanoseconds or even microsecond simulations in a day. And that brings us from a state where 10 years ago, all we could really look at on a day-to-day basis were simple things like water reorganization, which is extremely important in understanding molecular recognition, but really insufficient to really drive drug discovery projects. Once we start getting into the high nanosecond or microsecond timescales, we could start to see things like agonism and other conformationally related changes. We could see things like allostery, induced pockets and allosteric binding, and then look at the total relative binding free energy, including entropy and other effects, loop dynamics, domain breathing, and things along these timescales. So it is really a excellent time in the field of molecular simulations, and we feel that this is where simulations are really going to start making an impact in driving decisions in drug discovery, and not just helping to explain decisions. And that timing is because we could run a microsecond simulation 10 years ago. It just might have taken 6 months to run that calculation, even on a supercomputer, and therefore minimal impact in terms of drug discovery. As we're starting to get these calculations to complete on a daily basis, and we can complete hundreds of them across our hundreds of GPUs, we could really start to make big decisions that really drive our discovery process. So I'm not going to talk a lot about the platform foundations. There's a lot of details. I'd be happy to follow up with anybody after, but we've built out the best in class force field based on quantum mechanics. We run quantum mechanical calculations on every small molecule that we look up. We don't use, or that we run. We don't use lookup tables for any of these force field calculations. We derive our force field parameters from quantum mechanics from scratch in order to get the highest quality models. And from that, then we could start to look at binding free energies. We decompose the energetics onto atoms. And so we're not just producing a number, but we're producing insights as to why molecule A might be better than molecule B, what's happening on the protein, what's happening on the ligand, is there strain, desolvation, interactions, et cetera. We look at the water thermodynamics extensively. Water is really key in biomolecular recognition, and getting a visual representation of the water hotspots has been really crucial in many of our projects, and I'll show some of that in the STING project. We built out a very robust virtual screening platform that ultimately culminates in rigorous alchemical absolute binding free energy calculations, meaning that we compute not just the interactions like a typical docking program might do with a rigid receptor, but we have all the full protein flexibility, entropy is accounted for, waters, et cetera. So all of the parts of the system, of the relevant biological system, are contained. We've developed some enhanced sampling methodologies to explore these conformational hypotheses and this conformational modulation, which I'll talk a bit about at the end here, and the integration of biophysical data into our simulation algorithms. If we have time, we can talk about those a bit at the end, but I'm going to focus specifically on our small molecule STING agonist program because I think it's a great example of what we can accomplish using physics. So for people who are not familiar with STING, here's a representation here, a cartoon representation. It's a fairly large homodimeric protein that's central to the innate immune system. It's really considered to be the central regulator of the innate immune system. And upregulation of this target can cause inflammatory diseases, and downregulation can cause other problems on the other side of the spectrum. The idea here with our oncology program is that we can activate STING, we can trigger or spark the innate immune system, causing tumor infiltration and reduction of tumors. The thing that makes this protein so challenging is that it is large, and it undergoes a large-scale conformational change, going from this blue structure, which is the inactive structure or unbound structure, to the purple structure, which is the activated structure, which naturally binds cyclic dinucleotides. STING is a sensor of cyclic dinucleotides from aberrant DNA in the cell. So the binding site is large because it binds cyclic dinucleotides. Nucleotides are obviously polar and charged, and so that makes designing small molecules very challenging because you would typically then have to have a large charged molecule that is not cell permeable. And that's basically what's happened with the cyclic dinucleotides that are currently in the clinic for STING. Most organizations have gone after this target. They've taken the cyclic dinucleotides, the endogenous ligands, made a few changes to try to tweak the properties, but essentially you're still stuck with a cyclic dinucleotide that has poor properties. And the clinical trials thus far have all focused on injecting these CDNs directly into the tumor, which obviously limits the accessibility and tumor types that one could go after, and as well as doesn't provide systemic exposure to induce a robust innate immune response. So I'm going to show another movie here that'll summarize the work that we've done, and then I'll talk about each of the specific steps in sequence here. So again, we're running all atom molecular dynamics simulations where we treat thousands of water molecules around the protein in the most biologically relevant state that we can generate, and just trimming away those waters so we could see the underlying protein. What we'll first see is this conformational change going from the open to closed state around the cyclic dinucleotide. So our goal is to design a small molecule that can induce this conformational state, but by being a true small molecule with small molecule-like properties. And so we came up with this idea that we could develop a small molecule that dimerizes on itself in the context of the binding site. Now, this was just an idea. There was no activity initially, but we analyzed the water thermodynamics, and you could see here these red high energy water hotspots. And by building out into this region gave us our first binders. And that was really exciting, but we didn't have any cell signaling at that point. But we realized there was this pi-pi stacking interaction that was really driving this dimerization, and that's a complex thing to intuit. And so we developed a quantum mechanical algorithm to optimize the nature of this pi-pi stacking interaction, and that got us our first molecules with cell signaling. From there, we further built out into these high energy water hotspot regions and started to gain further binding affinity. But what we started to see is there was a disconnect between the binding affinity and the signaling. And here's one particular conformational change where a methionine comes into the site, wraps around this semi-hydrophobic group here, makes the binding tighter, but actually reduce the interferon signaling that we're looking for. So, we're a hypothesis-driven company. Sometimes hypotheses get refuted. We have to take a step back and we have to focus on what really matters here. And again, this was going beyond binding and focusing on conformational stabilization. And ultimately, we were able to drive to the conformation that you see here, which we feel is really the active signaling conformation that we're looking for, and really use that in the final stages of the project to drive toward our development candidate, which we are soon taking into the clinic. Here's just a quick picture, a zoom in of those water thermodynamics and how clearly you could see from where the ligand, where we initially had placed it in the binding site and with the yellowish color here, and a very clear visual path that helped our medicinal chemistry and design team to see how one would start to improve potency in this molecule. But again, there's a disconnect between binding and interferon signaling, and that's shown here in the top left figure, where interferon signaling is shown on the x-axis, cellular signal that you're looking for. On the y-axis, we're looking at binding, and you could see very little correlation here. Whereas if we look at the delta Tm, or the temperature shift using DSF thermostabilization, we could see that there's quite a nice correlation, and that's really one of the key insights that we gained as we were driving on this project, that it wasn't just about binding, but it was about some kind of conformational stabilization that's most related to the downstream signal. And what you could see here on this graph on the right is our compound SNX281, our clinical candidate, which has activity to the human protein, the human STING protein, and this is the delta Tm, or the thermal stabilization, but also to the most prevalent allele, the HAQ allele, as well as the efficacy models and the tox proteins of the tox models. So we could see that this was really pan-specific across all of the protein variants that we were looking for. Again, the wild type, the prevalent HAQ allele, as well as the efficacy and tox models. There was a program that was focused on STING a few decades back. The compound that went into the clinic was called DMXAA. And as you can see there, it had some stabilization on the mouse protein, but essentially nothing on the human STING protein. And ultimately that program failed, and we think we know why. It's because it wasn't stabilizing the protein that you needed to stabilize, obviously, the human. So here's an example of how development of custom tools is so essential in the context of challenging targets. And so if you search the entire PDB, the whole protein database, and look for any instance where a small molecule is interacting with itself in the binding site, you won't find anything. In the best case, there's one case where there's kind of an end-to-end interaction where things start to get close, but nothing where they're really interacting strongly. And so we had to develop custom tools specifically to handle this. That included custom docking algorithms where you're not just docking one molecule, but two copies of the molecule simultaneously. And then this tool here, this FSAP, which is a symmetry adapted perturbation theory approach using quantum mechanics, where we could specifically assess the nature of the pi-pi stacking interaction. And again, going beyond just computing a number, but producing visualizations that help the medicinal chemistry and design team understand not just what is better, but why something is better, and then how we can make it even better. And what we were looking at on the graph on the right is the correlation between those FSAP'd interaction energies and the actual underlying binding, which are quite good correlations. We also ran a lot of binding free energy predictions. We're typically looking at hundreds of molecules on a weekly basis in our design meetings and running these binding free energy predictions, which are essentially the most accurate in silico binding assay that's available. And we use that to prioritize or triage all of our ideas down into a manageable set that we send out for synthesis. So in this project, we were able to get to our development candidate in about 200 molecules synthesized and less than 2 years. So quite fast and relatively small number of compounds. What you're looking at in this graph here is the distribution of energies of the molecules that our RBFE, our relative binding free energy calculations, say are good, and the ones that we say are bad, good being in blue, bad being in red. And you could see the distribution, the blue distribution is clearly much better than the red distribution, and therefore really helping drive our decisions. Of course, none of these calculations are perfect. All models are models. It's just a question of whether they're useful. And this demonstrates very clearly that this is a useful model to help with compound selection. And then one more bit that was really useful for us is this integration of biophysical data. In this particular project, that was primarily NMR data, telling us some very specific details about conformational changes, such as that methionine flip that I showed earlier, as well as small angle X-ray scattering, or SAX, which helped us determine information about this open to closed conformational change associated with activity. More recently, we've started to integrate hydrogen deuterium exchange with mass spec, and we can include all of this experimental data included with our molecular dynamics simulations, or our molecular dynamics force field potential. And so this aggregation or combination of biophysical data and computational data helps our calculations in terms of accuracy, efficiency, as well as focusing our sampling on the biologically important motions in the system. And I should mention, crystallography, structural biology is central as well. In this project, we solved 25 high-resolution crystal structures down to about 1.4 angstrom resolution, and that was very enlightening and impactful in driving the project as well. All of the simulation doesn't stand on its own. It's really just a set of tools to help facilitate people to make better decisions, to gain insights into how the system works, and to drive our decision-making process. So this all comes together in our physics-driven design cycle, where ideas are proposed from our medicinal chemistry team and modelers, experienced drug discovery veterans, where they start to leverage the water thermodynamics, as I showed before, some of these atomic free energy decomposition, so we could see not just what is better or worse, but why things are better or worse and how things might be able to be improved, and this conformational modulation approach. All of these ideas that come out go into our design meetings, where we're looking at hundreds of molecules on a weekly basis with a combination of medicinal chemists, modelers, simulation leads, and biophysicists. And then we bring in the software engineering team as needed. And as I mentioned, we have about 30 software engineers and scientific developers that can be brought in as needed for specific project needs, like that quantum mechanical FSAPT approach, or in other targets, there's other challenges such as dealing with metal interactions or specific conformational changes. And all of these ideas then go into our simulation algorithms, where we're looking at binding free energies, typically hundreds of predictions on a weekly basis, as well as these conformational free energies. And then we iterate on that with experimental data, which ultimately leads to our development candidates. So just a little bit of data on our STING program. Here's just showing our interferon signaling, which is quite strong at low concentrations, especially relative to cyclic dinucleotides, our compound shown in blue, and cyclic dinucleotides shown in orange. And so you could see very strong signaling, more potent and tighter, and that's then leading to the downstream activity. And this is the culmination of all of those works. Just to summarize here, that we have a very strong anti-tumor response. A single dose, which is what we're seeing in the left side here, results in complete tumor regression, as compared to the untreated in the CT26 models, which are a PD-1 resistant tumor. And perhaps even more interestingly, there's a tumor memory. And so what we see here is that when we re-challenge the tumors or re-challenge the mice with the same tumor model, 3 months later, the tumors do not grow anymore. The body actually remembers that there was a problem, and it deals with it. So this is really exciting new area of research, really great demonstration of the general IO philosophy, the immuno-oncology philosophy, and STING as really this excellent target, central regulator of the innate immune system, perfect for what we're looking for. We're typically looking for genetically validated targets with a strong disease rationale, but that are chemically challenged, where people are stuck on creating a drug-like small molecule. And I think our STING program has demonstrated, in this very compelling data, shows that this is in fact possible, and these physics-driven approaches can really accelerate the drug discovery project process, and maybe not even to say accelerate, but enable us to drug targets that were previously considered to be undruggable. So with that, I want to talk a little bit about our future work, how we think about conformational modulation, and specifically this concept of conformational genetics. So the idea is that we could start with human disease-driven genetics. We can map that linear information onto a 3D structure, and then use that to assess how the protein changes, assess druggability, and ultimately use that to develop our target selection and design strategy. So I'm going to just have a couple slides on the approach that we use to do this. We've developed an enhanced sampling method called meta-EABF. That's a metadynamics approach with this extended adaptive biasing force. It's a very advanced method for simulations, and it really is what allows us to sample these large-scale motions and long time scales. And so what we do is we combine two advanced fields of sampling metadynamics with this adaptive biasing force to define a collective variable, and then accelerate the simulations to sample that collective variable. You could think of a collective variable as like a reaction coordinate. It's saying how to get from state A to state B. Obviously, in proteins and with protein dynamics, that's a very complex nonlinear path. And so we have to adaptively adjust this path to find the low energy pathway. And then once we've developed that, which can be very challenging and it can take many weeks or even months of computer simulations, but ultimately, once we solve that problem, we can start to then screen based on this collective variable, where we've identified the collective variable or the low energy path transition between two states. And then we could start to ask the question, what are the implications of a particular genetic modification, a gain of function or loss of function mutant? And additionally, how can we use this to drive our drug discovery projects and screen for compounds, not that just bind, but that go beyond binding into conformational modulation? And this is just to illustrate that this is a very nonlinear process, going from, for example, an open to closed conformational state. So let's just talk about a few examples. Here's one that we just use as a proof of concept, SHIP2, great example. And initially, Novartis did some fantastic work to identify an allosteric pocket through a large scale screen, and they've ultimately progressed that molecule into the clinic. What's interesting is when you look in the sequence space in terms of the gain of function mutations, you could see that there's some localized on the SH2 domain, shown here in green, and some localized on the phosphatase domain, shown here in red. But when you map these onto a three-dimensional structure, you could see they're all at the same interface here. And so what this tells us is there's this auto-regulatory functionality of the SH2 domain, and the mutations are probably influencing that. And so we developed a methodology where we can essentially pull on what I'm showing here in green, which is this regulatory SH2 domain, and compute the amount of work that it takes to dislodge this in the context of either the wild type, the gain of function mutations, or an allosteric inhibitor, which is represented here in orange. So I'm just going to show what one of those simulations looks like as we're pulling on this system with, in this particular case, a drug molecule here. This is a variation of SHP-099 that we designed. And what we could see here on the plots on the top right is that the gain of function curve, which is shown here in blue, in fact, introducing one of these gain of function mutations causes the lid or the regulatory domain to come off more readily. The wild type is next shown in here in gold. And as we start to optimize our compounds, we could see that it gets harder and harder to pull that domain off, meaning it's doing what we need it to do. And this really is something where one needs to go beyond binding. One could imagine if there's a small molecule that sticks to the bottom left of this purple protein, you could have an equally good binder, but it's not going to hold that domain in place. And that's where it really is important to understand the underlying biology and relate the simulation to the biophysics and ultimately to the assays. Here's just one more example of that. This is with NLRP3. This is a very interesting, fairly novel target also involved in inflammation. And what this does is it undergoes a conformational change from an inactive state, where you can see this purple domain here flips all the way to the opposite side. And this is just one copy of the NLRP3 protein. But this conformational change actually allows for a dimerization and ultimately this multimeric formation. It's an 11-mer that causes the downstream activation. And so panel C is called the inflammasome. One could imagine trying to inhibit this particular inflammasome state here, but it's very, very hard to imagine how one would do this with a small molecule. This is a very large system. How to impact this with a small molecule would be a challenge to think through. But instead, what we've done is stepped all the way back to the beginning here and said, can we, in the monomer, lock this or glue this conformation into this inactive state so it never has a chance to activate it in the first place? Again, so we developed a collective variable that helps us simulate this, and I'm going to show this how we're able to actually see transitions, many transitions going from this active to inactive conformation. And we've done this for the wild type protein as well as the 5 most prevalent gain of function mutants. And what we see is this genetic validation of the computational approach. So we think we have an understanding. Only the wild type system has an activation barrier going from this inactive conformation to the active conformation, which is what you would imagine. All of these gain of function mutants reduce that barrier, which then therefore explains the activation process and the inflammation response. And really this then leads us to a hypothesis about how the system works and how one could go about drugging it. So just based on time, I'm going to wrap things up here. Again, be more than happy to talk with people offline or after the meeting about the details of the platform or anything else. Silicon Therapeutics is the only fully integrated physics-driven drug discovery company, the only ones that really have everything from chip to clinic. We've built an industry-leading platform for molecular simulations and drug discovery. The development of custom apps is really key for drugging challenging targets, and I think that was demonstrated on the STING program, and we've seen this in our other internal programs as well. The integration of biophysical data enables more accurate and biologically relevant simulations, and our small molecule STING agonist program demonstrates the value that physics-driven simulations can bring to tough projects. And with that, I'd like to thank you all and leave it here.
Wonderful. Thank you again, Dr. Woody, for your amazing presentation. There are a few questions for you. One is from Mr. Hugo Gagnon, CEO of PhenoSwitch Bioscience. His first question for you, Doctor, is how hard is it to find or how available are targets like STING with the perfect set of information?
Yeah, so that's one of our biggest challenges, absolutely, is finding those right targets. The targets that are genetically validated, biologically, very interesting, and a path from the target to the clinic, but also chemically challenging. So it's certainly not every target, but I think for most people in the field right now, they'll agree that there's a lot of them. And most of these more interesting targets, especially as you start talking about intracellular signaling proteins, this is a fairly significant population. I showed one phosphatase, just the SHP2 example, but there's many other phosphatases that have different regulatory domains that we feel that this approach could be highly valuable for an allosteric phosphatase program. these signaling proteins in inflammation as well. So again, not that they're everywhere. It takes a lot of work, and that's, I'd say, one of our biggest efforts is generally in target selection, and we choose it not just based on the genetics, but again, a lot of simulations and really trying to find that sweet spot that balances the genetics and biology with the simulations and our insights. So they're there, but it takes work to find them. And I'd say, just going through the recent literature, if you look at a good majority of the hottest targets, I'd say they fall into this category.
Wonderful. And the second part of this question is, from chip to clinic, what is the biggest pitfall and difficulties right now for going from computed best candidate to in vivo working molecule?
Yeah, so that's an excellent question, and it really is. That is the challenge. Is actually that connection between what we're simulating. So we simulate a protein that we think is the biologically relevant system, but biology is complicated, and understanding which assays to develop that connect our simulations to binding to cellular signaling, is where the greatest challenges and most of our effort is spent in the drug discovery programs. We're obviously developing new computational methods, and that's challenging as well. But I think we have clear insights into how we're going to advance those programs. But this connection between protein simulations, binding assays, and cellular signaling is challenging because every target is unique, and it's something you can't just develop a workflow or some automated program to do. I mean, we're talking about hard biology. We're talking about reading a lot of papers, really trying to understand what is the relevant construct, what are the relevant conditions, are there any cofactors involved, what's the signaling pathway. So that is the greatest challenge by far, and what we spend most of our time on, and what keeps us up at night.
Wonderful. And there's a question from Merwan. He said, "Great presentation, Dr. Sherman." His first question for you is, for small molecule force fields, do you mean you derive bonded and non-bonded parameters from both QM in a de novo way? If yes, in practice, how many molecules can you afford per day, and which QM method do you use, like DFT, wave function based, semi-empirical, etc.?
Yep. So we do use DFT and typically B3-LIP for those that are familiar with the field. We have a GPU-enabled QM program, so it takes us about an hour to fully parameterize a molecule, and that means doing full torsional scans and geometry optimizations and electrostatic potential fitting. So this is only really possible because we're running quantum mechanics on a GPU, and that's the only way that we could do this fast enough to actually not be a rate-limiting step in our process. As far as the bonded and other parameters, those are things that we don't do on an every molecule basis. We do a lot of those up front, and then that just provides the foundation for the force field. But it's the torsional parameters and the charges that we update on every molecule on the fly.
Wonderful. The second part of Marwan's question is, for the adaptive sampling, do you believe the method can be used to sample experimentally validated protein conformations?
Yeah, absolutely. And in fact, we've demonstrated this in a few cases where we've been able to make predictions of states that either we or others have ultimately validated through crystal structures and through NMR, for example. But we do generate a lot of other states, and it's hard to say specifically whether those are validated, because like you said, experimentally we can't capture these states, but we can gain insights and using other biophysical methods. So for example, we use a lot of hydrogen deuterium exchange mass spec to understand which parts of the protein are getting more buried or more exposed, and that can help to validate some of these models where we're generating states that are not crystallizable because they're either higher energy, less populated states, or require a ligand to induce those states. But the HDX can help give us insights into that. NMR as well can help give us insights into some of these more transient states. And so the work that we have been doing has validated a lot of these states that we've generated. And we also use the hydrogen deuterium exchange and NMR data to help guide our simulations so that we can do that more accurately and efficiently.
So is there any other questions for Dr. Woody Sherman? And as you mentioned, if you have any other questions offline, you're more than welcome to connect with you to answer regarding this program. And once again, Dr. Woody, thank you again for your wonderful presentation.
Great. Yep. Thank you all.